Today's session for OICR Monday is titled Immuno-oncology Moving Beyond the Asymptote, and I'll tell you why we sort of chose that term asymptote. It's a mathematical term, but I think it has some connotations here, and we have some really fantastic speakers: Dr. Marcus Butler, Masroor Bayati, Jonathan Bramson, and as I said, I'll introduce them more thoroughly when they give their talks, and then I'm the moderator. So, immuno-oncology and immunotherapy is really kind of aiming at this sort of idealistic sort of situation, which is sort of shown here in this so-called cancer immunity cycle that was originally published by Ira Mellman and Dan Chen from Genentech a number of years ago. And we all sort of show this, and that is we all want to have this ideal situation where we have therapies or de novo dying and killing of tumor cells, and these release antigens that then are picked up and presented by antigen presenting cells, which, the best ones are dendritic cells. They activate a T cell response. These T cells then expand, and then they usually in the local draining lymph node of the tumor, then they traffic back to the tumor, kill the tumor. And then of course the cycle continues because then these T cells cause more damage. And so this cycle actually represents a whole opportunity for a whole bunch of combination therapies using a whole bunch of different modalities to sort of hit various mechanisms of this cycle where there are gaps in different types of cancers and different types of patients to then more thoroughly activate a T cell response. And of course, this whole notion of immunogenic cell death, killing the tumor in the right way, is becoming a huge question because there are right ways and wrong ways to kill a tumor cell. So that's very important. And of course, this is being done in the backdrop of immense tumor heterogeneity in space and time, and also the role of germline variations in different people, where different people are simply hardwired to mount immune responses in different ways. And if anything, we've learned that from the COVID-19 pandemic, where some people die of COVID and others, they don't even know they have COVID. So this is going to be a huge area that we need to still work on. And this is where the asymptote comes in. Right now, as some of the speakers will also tell you, we made tremendous inroads, from the early 20th century now into the 21st century, where we had chemo radiation therapy come in, we had then targeted therapies, and then of course in the 2000s we had immunotherapies finally taking off. And each of these sort of horizon marking kind of shifts in our way of treating patients led to these increases in improvement in patient survival and benefit. And of course, when the checkpoint blockade inhibitors, anti-CTLA-4 and anti-PD-1 came on, this sort of then spurred this sort of hump and rise. But we're in this point right now where we've activated and learned to activate the immune system really, really well. But we're sort of at a conundrum, like, where do we go from here? Like, how do we go beyond anti-PD-1 that has an anti-CTLA-4 and some of these other major checkpoint blockade molecules that really activate the immune system? And although I didn't want to see this as negative, as it's really asymptotic, but we're really reaching a situation where are we going to go here? Like, some of the things we're adding on to these checkpoint blockades are only getting minimal incremental increases in tumor control and survival. And so we're really at a point where we really need to do better and really understand the mechanisms of action, do better precision medicine in order to do this. And so, and this is the problem, is the complexity. There's like a plethora of thousands of different possible combinations. How do you do all these trials? Which one do you test and when and how? So this is the major problem right now. We're dealing in immuno-oncology. And of course, we're dealing with a whole array of different types of therapies, right? Where do we go? Like, do we use vaccines? When do we use vaccines? Do we do cell therapy? When do we use both? Do we combine? How do we combine radiotherapy with immunotherapy, et cetera, et cetera? So it's becoming very, very complex because we have a whole bunch of different types of therapies to choose from on top of this. And on top of this, we're now in an era of molecularly targeted small molecule drugs, targeting everywhere from BRAF to RAS to EGFR to c-Met to VEGF, et cetera, et cetera. And the first order of approximation will be to treat patients based on molecular profiling and mutational analysis with these really ever exceedingly or increasingly specific, potent targeted drugs, small molecule drug. The question is, how do we position IO therapies in this small molecule cancer drug space? Where do we go? how do we control the negative and positive effects of these drugs? Because these drugs are pathway inhibitors, not molecule inhibitors, and they're also expressed in immune cells. And so many of these targeted therapies can actually have negative effects on the immune system. But how do we deal with that? And then the other question is, cost of drugs, right? Cost of drug development. We need to have better paradigms of drug development, more different clinical trials, better clinical trial paradigms, better sort of clinical readouts to inform the drug discovery, drug development, moving from these different phases of development into regulatory. It takes about 10 to 11 years, $2 billion or more, and less than 12% of drugs make it. There's got to be a better way. And immunotherapy is suffering now from the same problem. And then accelerated approval is another problem. Many drugs are IO agents are being approved on accelerated approvals, and many of these accelerated approval drugs are not showing any benefit in terms of increasing survival, as shown here. And on top of that, many drugs that are not showing increases in survival, up to 33%, are not withdrawn from the market. We're still using them. So there's a lot of junk out there. And unfortunately, there's some junk out there in immunotherapy as well. So so there's a couple of questions that I'll be, addressing the panel. I'm not going to go through them now, but I'm going to, I think, I'll stop sharing and I'll introduce the first speaker, and that is Marcus. So Marcus, if you want to share your screen, or I don't know who's sharing the screen for you. So Marcus is a physician investigator who's done a lot of work in immunotherapy, especially cell therapy. He was trained actually at Dana-Farber, came to Toronto. He's a member of the medical oncology staff at Princess Margaret. He's a melanoma and skin medical oncologist and site lead for PMH's immunomonitoring team. And he focuses on immunological impact of cancer immunotherapies. And he's also the medical oncology chair for the melanoma site for the Canadian Cancer Trials Group. So Marcus, welcome and thanks for joining us.
Okay, great, thanks.
Yes.
All right, so these are my disclosures. So thanks, Laszlo, for the great introduction. So this is an earlier version of the cycle of immunotherapy concept that Laszlo started off with, which brings up the point that there are multiple elements along the way of developing an immune response where you can basically the cancer disrupts that immune response and then results in a successful tumor, whereas immune checkpoint inhibitors and other immunotherapies are able to interfere with what the cancer does in order to induce clinical benefit for patients. So to just sort of move forward for considering time limit that we have, basically I wanted to focus my talk as kind of a very 30,000 foot, where are we with immunotherapy? So with immune checkpoint inhibitors, we've had a lot of success. A lot of the success and many of the drugs approved are basically the same target, anti-PD-1, anti-PD-L1. There's one drug that's been approved for anti-CTLA-4, but up until recently, just last week, those were the two pathways that molecules that were successfully approved for the treatment of patients with cancer. And melanoma is a great model for immunotherapy in that it's highly successful, and yet we see all of the challenges of immunotherapy with immune checkpoint inhibitors in terms of mechanisms of resistance and that develop for patients. So even though it's highly successful in many patients with melanoma, there still are a good chunk of patients who have primary resistance that don't respond at all to immune therapy. These patients may have a lack of a nascent immune response, so the checkpoint inhibitors are not able to result in clinical benefit. And then also patients do develop acquired resistance using multiple different types of mechanisms, such as beta-2 microglobulin loss or other mechanisms of resistance that results in inability of the tumor to be the target of therapy. But in cancer, we have 10 years ago, we had all this excitement about success in patients. And in general, the concept that patients that have an inflamed tumor are more responsive to immune checkpoint inhibitors holds true, where many patients who have less inflamed tumors just don't respond to these immune checkpoint inhibitors. So the low-hanging fruit has basically been we've been able to develop a lot of therapies, but it's basically low-hanging fruit where we're dealing with resistance mechanisms for the remainder of patients. In terms of the types of immune mechanisms of resistance, patients may have T-cell suppression, immunosuppression with multiple additional immune checkpoint inhibitors present within the tumor microenvironment. There may be poor antigen presentation so that T-cells just aren't able to recognize the cancer, barriers to T-cell infiltration, and a lack of T-cell response where T-cells are not able to recognize antigens that may still be present. So in order to deal with these mechanisms, there are multiple approaches to look at combination therapies with multiple immune checkpoint blockade mechanisms. And up until last week, we had no successful therapy, although now anti-LAG3 combined with anti-PD-1 has approval in the US, so there's at least some success. But that's not without a lot of additional combination therapies that have basically been unsuccessful. Poor antigen presentation, loss of MHC, there are various strategies to try to make a more immunogenic tumor microenvironment, and a lot of the work is focusing on STING agonists, anti-VEGF therapies, CD40 to address that. In terms of barriers to T-cell infiltration, anti-VEGF therapy targeting the Wnt beta-catenin pathway is an area of continued interest, and there are clinical trials. And then for lack of T-cell response, vaccination strategies with oncolytic viruses or epigenetic strategies, as well as synthetic immunity where T-cells are engineered to then be transferred as a therapy. So there's really been a huge number of trials to look at various combinations. It's kind of shocking at how many different trials were present. This is back in 2017. And yet we still, despite all of those trials, have Ipi-Nivo has been approved for the last 5 years plus. And then now we have LAG3 that's just been approved, as well as cell therapies with CAR T, hopefully soon a TCR-T therapies will be approved, as well as bispecific T-cell engagers. But basically, all of the clinical trials that have been conducted, except for a few attempts to try to use biomarkers to treat patients, essentially were really, for most, at least ready for prime time. The successful approaches are treating all patients essentially the same. We do not have BRAF mutation or a particular other molecular target that we're able to sort patients appropriately for various therapies. So as a result, there's a lot of overtreatment as well as undertreatment of patients, where some patients probably don't need 2 years of anti-PD-1 therapies, where other patients need a more robust approach. One of the strategies that I think shows a lot of promise in melanoma as well as other cancers is to do a neoadjuvant clinical trials and also moving into standard of care. In melanoma, there are several examples of resectable stage 3 melanoma patients who receive neoadjuvant therapy. And it turns out that the ability to have a path CR or near path CR is associated with much better outcomes compared to patients who have partial responses or no CR. So that we are likely to be able to de-escalate treatment in patients who have, after just 2 doses of immunotherapy drug, have a complete response pathologically compared to other patients who do not. And there are trials such as the NADINA trial, where patients who have path-CRs are then receive no further treatment or monitored, whereas other patients are who unfortunately do have evidence of progression or non-path CR are dose intensified in terms of therapy. Now, I think synthetic immunotherapy is probably the next wave of treatment for patients with melanomas and other solid tumors, where we really have kind of maxed out, I think, what we're going to see with immune checkpoint inhibitors. So this is the approach where vaccines or engineered T cells are used to treat patients, as well as bispecific molecules or expanding tumor-infiltrating lymphocytes. So for cell therapies, there are three main types: tumor-infiltrating lymphocytes, taking precursor endogenous T cells, expanding them through manipulation in vitro, and then infusing, and then gene-engineered products such as CAR T or TCR-T. With TIL in patients who otherwise have very little chance to respond to therapy, various companies are seeing efficacy in this patient population. I think one of the challenges that the pharma has with this type of therapy is the fact that the patient groups are so heterogeneous, and really being able to satisfy regulators for clinical benefit is a challenge. In terms of gene-engineered products, there are CAR T as well as TCR-T approaches. For CAR T, lymphomas, myeloma, leukemias are being targeted with CD19 CARs with great success. But this has, for CAR T, it really only targets the tip of the iceberg in terms of tumor antigens that are present within patient's tumor, whereas other approaches such as T-cell receptor engineered products can target many antigens that are basically silent to the immune system without presentation by MHC. So the NY-ESO-1 as well as MAGE-A4 and HPV-16 are being shown to have promising results, with MAGE-A4 and NY-ESO-1 in sarcoma likely to result in approvals in the next few years or so. There was also a recent approval for an immunotherapy against uveal melanoma, a rare type of melanoma, which is quite distinct from typical cutaneous melanoma. Uveal melanoma has a limited number of mutations, although several that they hold in common, and has a low mutational burden, as well as low expression of PD-L1 in the tumor microenvironment. So uveal melanoma was the target of a drug called tebentafusp, which is a bispecific agent that targets GP100, which is processed and presented in the context of HLA, and then activates T cells through an anti-CD3 receptor. What this agent does is recruits T cells to the microenvironment. There's, you can see, a lymphopenia immediately after treatment due to a cytokine release, and then trafficking of T cells into the tumor. In the clinical trial, a single-arm study was actually did not meet endpoint with a very low response rate. However, there were a good number of patients who had better than expected survival. And in a randomized study comparing best investigator choice versus treatment, there is an overall survival benefit. But what I'd like to make sure this audience is aware of is that RECIST response was a very poor surrogate for measuring benefit of the drug. So it was a very low response rate, and actually the patients with progressive disease as their best overall response had a substantially increased survival compared to patients who received investigator choice. Whereas if you had a response to immune checkpoint inhibitor or to tebentafusp, the overall survival was similar for both of those groups. A better surrogate for benefit is ctDNA. As I mentioned, there a handful of mutations that are common in uveal melanoma, and therefore a panel testing was developed by Natera, and then has shown that for patients who have significant reduction in ctDNA or clearance of ctDNA, had much higher overall survival benefit compared to patients with no reduction. So in summary, there's been an impressive investment in multiple immune modulators. A lot of those agents are now sitting on a shelf because they were safe, but not effective. And I think the companies don't really know what to do with a lot of these drugs. There are a lot of obstacles for combination trials because many companies have developed their drugs and they're now sitting in silos, not being combined with other agents. We have the issue of overtreatment and undertreatment in patients, and there really is a lack of biomarkers to help guide us with therapy in the clinic. Even for LAG3 expression, it's not in melanoma, it's not a one-to-one, oh, if you have LAG3 expression in the microenvironment, then you'll benefit from relatlimab. It's not the case. There have been a lot of success in cell therapies, persistence, graft function, as well as tumor microenvironment issues remain a challenge for cell therapies, and certainly developing cell therapies in solid tumors is the next focus that the field is looking at. Now, in terms of surrogate measures of benefit, I think it's very interesting that you have this rare cancer. PFS is basically a poor predictor for overall survival benefit, but I think that is important, and we often, especially regulators, get so focused on RECIST as a surrogate endpoint that leads to accelerated approval. It could be that we're making some choices with drugs based on response rate that is not really meeting our needs. And as Laszlo pointed out, many of these accelerated approvals don't result in clear overall survival benefit. So maybe radiomics or ctDNA or other strategies to try to measure benefit of these therapies will be the next step, but I think that that's an area that warrants a lot of focus in the future. So thanks very much.
So the next speaker I'd like to introduce is Masroor Bayati. Welcome, Masroor, and it's great to have a trainee and a student or postdoc that joins us here, and it's a great opportunity, and we hope to have many more of our students and postdocs do these sessions, so welcome. Masroor is a PhD student in medical biophysics in Reimand's lab. Yuri is a PI at OICR in Computational Biology, and his research is focused on the application of machine learning and identification of novel prognostic cancer biomarkers, leveraging multi-omics data. And I think you're currently working on prognostic value interaction between cancer genetic events and the immune cell composition. And I've seen some of your exciting work in this area coming out of Yuri's lab too. So welcome, and looking forward to your talk.
Thanks, Laszlo, for a great introduction, and hello, everyone. And I'm very thrilled to be in this panel. Such an honor for me. And as suggested by the outline of this program, I'm going to give some motivation of application of AI and what position it has in this vast realm of immuno-oncology. So I found it useful if I talk more about my own project that we pursue in Reimand Lab that kind of motivates the use of AI as a searching tool for novel biomarkers in cancer. So my agenda is that I will talk about the artificial intelligence and cancer biomarker discovery when they meet each other, what kind of challenges we face. And then after that, I'll talk about my own project, which is more about integrating two data modalities that we have in immuno-oncology, which is tumor genomics for genetic events, both in terms of driver mutations and chromosomal aberrations, and the immune signatures that we have, more specifically the composition of the tumor microenvironment. And this search for novel biomarker discoveries when we combine these two data modalities using AI. And then I will give some closing remarks for conclusions. So first of all, large consortiums such as TCGA have profiled about 10,000 tumors across more than 30 cancer types using multiple molecular and clinical platforms. And this specifically provides a good opportunity for artificial intelligence techniques to search for novel prognostic and predictive biomarkers for cancer. So I really want to emphasize on the fact that AI is more of a searching tool than a artificial intelligence, as its names might suggest. And in fact, AI works, as I said, as a searching tool, and it helps addressing questions such as what are the distinctive subgroups in a population, or what are the molecular factors underlying these differences, and what are the factors that could predict clinical outcomes. However, as opposed to very successful outcomes coming from application of AI in other scientific fields such as imaging, network analysis, social media, when it comes to biomedical applications, AI faces a lot of challenges. The most important challenge is data size. In terms of data size, especially when it comes to patient tumor data, the number of samples is relatively low compared to dimensionality of the data. Just focusing on gene expression, you can imagine we have thousands of features, each of which being a gene, but the number of samples, which are the number of patients, are typically around 100 or hundreds of patients. So this is what we call in machine learning and AI the curse of dimensionality, which really puts machine learning or an AI in difficulties to get inferences from the data. On the other hand, within each cohort, different platforms are applied in a low overlapping manner. That is, for example, not all patients that are profiled for somatic mutations are also profiled for RNA expression. Additionally, any type of profile is pre-processed in different cohorts using different computational pipelines. And all of these issues limit the generalizability of the AI techniques for biomarker discovery. On top of all these, AI algorithms come with a trade-off where more complex algorithms often are less intuitive and less interpretable, and they tend to be like a black box. And this limits the application of AI in the clinic specifically. So our work in Reimand Lab for biomarker discovery is more focused on genomics and immune microenvironment of tumors. As illustrated on the left, tumors develop via accumulation of driver mutations, and different driver events at different time points give rise to heterogeneous subclones of cancer. So driver mutations alone have been widely used in clinic to define distinctive subgroups of patients and to guide therapeutics. However, we all know that tumor evolution is not independent of tumor microenvironment. The cartoon on the right depicts different phases of tumorigenesis and the interactions between tumors and immune system. So in early stages of tumorigenesis, immune surveillance might eliminate the majority of tumor cells, but as tumors evolve through the driver mutations, they acquire mechanisms to recruit immunosuppressive cells and to develop characteristics to evade anti-tumor responses. Indeed, recent studies have shown that composition of tumor microenvironment specifically has prognostic and predictive value. So here you see an AI workflow named CyberSort, which was published about 6 years ago in Nature, which measures the proportion of immune cells in bulk tumors by computationally deconvolving the bulk RNA-seq profiles. This is one very important application of AI in immuno-oncology that we see. So briefly, the analysis is based on a signature gene matrix representing the unique expression pattern of different immune cells across a set of signature genes. And the algorithm tries to reconstruct the RNA profiles of tumors in terms of linear combination of these signatures. Then the coefficients of these linear combinations are considered as the proportion of each cell type. So I want to, at this point, talk about my own project, which is, as I said, the integration of tumor genomics and immune signatures as a novel prognostic biomarker discovery pipeline. So our hypothesis in this project is that the information of genomic aberrations and the immune composition of tumor microenvironment can develop a search space for novel biomarkers. So we characterize the TCGA tumors with a set of genetic and immune features. As you can see in this diagram, we have mutation features indicating non-silent mutations, the status of known cancer driver genes. And on top of that, we have the immune features, which is the result of CyberSort applied to TCGA cohorts, which are the binary variables indicating high or low infiltration of certain immune cells in different tumors. And then we ask this question, what subset of these features or combinations of these features that you can see in the middle are more indicative of survival information of the patients? And we attempted to find these, let's say, prognostic features using the following pipeline. So for each cancer cohort, a 5-fold cross-validation is performed. So within 80% subset of the data, we run 1,000 iterations of bootstrap subsampling. For each subsample, we fed a multivariate Cox proportional hazards model, which was regularized by elastic net method. So this process will give us a minimal set of input features that optimally explain the variation in survival outcomes. And within each cross-validation, we have the chance that, to see that how much the detected prognostic features are generalizable to the unseen portion of the data. So this is the results of the pipeline for several cohorts of TCGA. We applied the pipeline to not only the combination of mutations and immune features, but also to combination of chromosomal copy number gains and losses with immune features. On the vertical axis, we have the immune features, which are the around 20 major subsets of the immune cells in the peripheral blood. And on the horizontal axis, we have the genomic events or the genomic event features, and each color indicates different cancer cohort. So each colored point indicates that when the corresponding pair of the immune and genomic feature occur and are combined in the corresponding cohort, it would divide the patients into distinct survival subgroups. So this plot had two important messages for us. First is that the copy number events provide relatively larger search space for biomarker discovery compared to mutational status of driver genes. And the second message is the heterogeneity of cancers in these prognostic combinations. That is, not a single combination was found prognostic in more than one cancer cohort. So we have a closer look into one of these prognostic combinations that we found in liver cancer cohort. So these are Kaplan-Meier plots. I guess nearly all of you are familiar to that, but briefly, it shows estimates of survival probability at different follow-up time points. And each line, each colored line here corresponds to a subpopulation of the cohort. So intuitively, when a line is pushed toward the lower values, it means that the corresponding subpopulation have lower chance of survival or worse prognosis. And this enforces the assumption that that group are potentially a distinctive clinical subgroup. And this is what we see and observe in TCGA liver cancer cohort. Initially, when we divide the patients based on high or low infiltration of monocytes, the Kaplan-Meier plot on the left shows no sign of survival difference. When the TP53 mutation or the status is taken as the dividing feature, the Kaplan-Meier plot is also showing not a significant difference between the subpopulations. But when we combine these two features, we will get the third Kaplan-Meier plot on the right, where the patients are divided into 4 groups. And one of these groups has very distinctive survival outcome, as you can see in the purple line here. And this corresponds to high infiltration of monocytes and TP53 mutation, actually. So interestingly, when we looked into an entirely new liver cancer dataset, which was a Japanese cohort from ICGC, the same survival trend was observed. That is, the patients with high infiltration of monocytes and TP53 mutation together had significantly worse survival outcome compared to the rest of the cohort. Such observations suggest that prognostic information in immune and genomic features, especially in their combination, might be generalizable and clinically valuable. So lastly, to explore the functional associations, we performed differential agent expression analysis based on the high risk and low risk stratification of liver tumors using this monocytes and TP53 combination. So interestingly, several known cancer driver genes were found towards the extremes of this volcano plot in the middle, suggesting their dysregulation. We also performed pathway enrichment analysis using the differentially expressed genes. And here in the enrichment map on the right, you see the pathways indicated as circles, and the connected pathways that share several genes are connected as lines here between these circles. And the different pathways are positioned in different functional themes here. So interestingly, we see that when these risk groups are compared, several cancer-related pathways are overrepresented, such as DNA repair, cell cycle, TP53 regulation. It is also very interesting to us that we see pathways related to collagen formation, as you can see here, that are overrepresented. And this observation somehow suggests that monocyte infiltration on one hand, TP53 mutation on the other hand, may be functionally connected to liver fibrosis and hepatocellular carcinoma through collagen-related pathways. So in conclusion, I just wanted to emphasize that AI helps narrowing down the search space of the biomarkers and guides new hypotheses. And specifically about my project, we were somehow successful to show that combining driver mutations and immune infiltration signatures in form of novel prognostic features and a prognostic in a cancer-specific manner, reflecting cancer heterogeneity, and they potentially improve on prognostic utility of each individual data modalities, which are mutations and immune features. And they potentially provide new functional insights, as we saw in the pathway analysis. So with that, I want to thank my committee and my teammates in Reimand Lab. And thank you all for joining this presentation.
Great, thank you, Masroor. That was a really polished, really good presentation. I came out of it understanding exactly what you're doing. Congratulations. Wow. Congratulations, Yuri, too, as well. Great, excellent work. I want to follow up now and with our last speaker, which many of you know, Jonathan Bramson. Jonathan is quite a well-known immuno-oncologist and very talented researcher. He's launched his own biotech company, Triumvira, which is really has a revolutionized sort of approach to sort of redirected antigen-specific T-cell therapy. And he's the Vice Dean of Research at the Faculty of Health Sciences and professor in the Department of Medicine at McMaster University, just down the street from us. And he's the John Bienenstock Chair in Molecular Medicine, and he's really focused on cell therapy and cells as drugs, as I just mentioned. And his lab is developing methods to manufacture leukocyte or immune cell drugs, optimizing anti-tumor activity, and using combination of genetic engineering, synthetic biology, and other chemical biology approaches. And he's also dabbling in small molecule drugs. He's got a CTIP grant to develop a CBL-B inhibitor, which is showing some very interesting, exciting data. So welcome, Jonathan. I'm really looking forward to your presentation.
So it is true that we have begun to dabble in small molecule drugs to modulate T-cell function, but those works are still very early on. So I thought I would take a different spin and talk to you about a less well-known area where small molecule drugs are still being employed to modulate and enhance T-cell therapies, because this is something you probably haven't heard of. And I thought it'd be fun to talk about. So yeah, I'll be talking primarily about the use of T cells as drugs. And so in terms of why, well, T cells are actually pretty handy cells to use as drugs because they're really easy to grow. So while many cells are very difficult to grow outside a human, T cells grow like stink. You can grow billions and billions of T cells with very little effort inside of a couple of weeks. Another valuable thing about T cells is they're readily engineered, so we can make them do lots of different things by either forcing the expression of certain genes or editing out the expression of certain genes, or as many of us do, doing both at the same time. What's nice about T cells as well is you can get them from third-party donors, so you can derive them from induced pluripotent stem cells. You can also use non-canonical populations of T cells, and this allows you to generate off-the-shelf products that ultimately may be of high value in a very broad spectrum of utilities. And most importantly, although we'll be talking about oncology today, T cells can be used to treat probably any illness because of their fundamental involvement in most aspects of human pathophysiology. Let's see if I can get the slide to move. So the process of treating patients with T cell drugs has been termed adoptive therapy, and I like to refer to it as a virtuous cycle of adoptive therapy. This began originally with the use of autologous products, so materials that were derived from individuals. So in this case, for example, a cancer patient would have their blood removed, modified, expanded inside of a dish, and then reinfused in the patient. But more recently, we've figured out how to make this work from third-party donors. So now that cycle isn't so much of a cycle as much as a direct path, where we are now manufacturing T cells from individuals who are healthy or from healthy pluripotent stem cells, and still following that same path afterwards where they are expanded and engineered and ultimately infused into the patient as a drug. We know now, and Marcus presented this information in his presentation, that in fact we can exploit T cells to cure cancers. And if you have enough T cells and they have the right specificity, this battle can work in favor of the oncologist. However, tumor-specific T cells are quite rare and therefore challenging to find in nature. So the solution that folks like myself have come up with is to say, well, if you can't find it naturally, just create it. And we've taken to engineering T cells to give them the specificity against tumor that we want. To help you understand how T cell engineering works, I thought I would show you a schematic of how the T cell sees the world. So on the tumor, you'll have a target that is composed of the major histocompatibility complex with a peptide derived from either an intracellular or surface protein. And this is called the MHC peptide. And it is seen by this complex series of proteins on the T cell side. The proteins in green are known as the T cell receptor. And there's actually 8 proteins that form this complex that ultimately engages the MHC peptide. And this complex is exquisitely selective for both the MHC and the peptides, so that only the T cells in my body can see my MHC with my peptides, and the T cells in your body see the MHC with your peptides. And although we're learning a lot about how these T cell receptors interact, there's still a bit of a challenge to work with on the engineering side. Nevertheless, as Marcus pointed out, we've learned that if we clone out the alpha and beta chains of the T cell receptor and express them ectopically in a separate T cell, these 2 chains alone can confer specificity to that T cell. So it is possible to manufacture large numbers of T cells simply by engineering them with T cell receptor alpha and beta chains. And this is what Marcus referred to as the TCRTs. This works. We've got some nice clinical data now emerging from some of the TCRTs, as Marcus pointed out. But as I mentioned, the MHC peptide complex is very patient specific. And so ultimately, some of the challenges that face the TCRT approach is that you have to have a large number of TCRs cloned and ready to go in order to capture the full breadth of all HLA. Now, having said that, that's not stopping folks moving forward, especially as they learn more about promiscuous TCRs and epitopes. So folks have decided to combine immunology with synthetic biology and recreate nature in the form of synthetic receptors that would identify tumor antigens. And the most well-known one is one referred to as the chimeric antigen receptor, or a CAR. And what I'm showing you here is the structure of a CAR in very loose terms, where one component of the TCR, known as the zeta chain, has been fused to a series of proteins that ultimately converge upon a ligand bind domain that selects tumor antigen. So why was the zeta chain selected for this? The zeta chain contains 3 of what's called immunotyrosine-based activation motifs, or ITAMs, and these are the critical tyrosines on the TCR that lead to signal transduction. And in the early days of characterizing the TCR, it was discovered that if you simply express these three ITAMs in a T cell and trigger a ligation event where they come together as a multimer, you'll be able to activate that cell. And that early molecular biology understanding eventually led to what's called this chimeric antigen receptor. And what's interesting about the CARs is because they bind directly to tumor targets, they do so in an HLA-independent fashion, which allows you to use a single CAR across all individuals. And so for folks like myself, we find that compelling because now you don't have to worry about matching the major histocompatibility complex, also known as human leukocyte antigen. So while this may sound like a fantastic idea that you could take T cells and engineer them and reinfuse them into a patient, this is a real thing. And in fact, there are 5 approved therapies currently marketed in the US, 4 of which are marketed in Canada. They target more than one protein, so Kymriah, Breyanzi, and Yescarta all target CD19, while Carvykti and Abecma, I'm not sure how they pronounce that, ide-cel, target BCMA. And there's many, many more to come. So there are many targets that have been identified that seem to have good therapeutic indices, and we're seeing very nice results from clinical trials. By and large, this has all been limited to liquid tumors, leukemias, lymphomas, and myeloma, but there's no reason to think we won't be able to get past that area. So really exciting days and looking forward to seeing great things come down the pipe. Oops, sorry about my friends. Let's go back. Wrong button. So inspired by the success of chimeric antigen receptors and the knowledge of TCR signaling, groups such as myself have come up with a variety of different mechanisms to redirect T cells against tumor targets. So I have taken to referring to the field as being synthetic antigen receptors as opposed to chimeric antigen receptors because they all have very different structures. Some of them are TCR independent, such as the CAR-Ts, as well as the DAP12 based synthetic antigen receptors, where the signaling is mediated through a separate receptor altogether. And then you've got the TCR-centric receptors, such as the one my lab has developed, the TAC, all of which attempt to redirect natural signaling. And you can read the papers and they'll describe the various attributes of these receptors. They all have their good sides and their bad sides. Ultimately, as we learn more about the clinical requirements for efficacy, we'll make decisions about which synthetic antigen receptor works best in which field. But they all suffer from one limitation, and that one limitation is their antigen specificity. And so this is an area where small molecules are really quite attractive, and I'm going to talk to you about that in a moment. So if you notice, I refer to the tumor binding domain in this case as this blue little horseshoe, I guess. And you'll notice that each and every one of these synthetic antigen receptors has only one tumor binding domain, which means you're making T cells reactive against only one tumor target. And we know that when you target only one thing in tumors, whether it's with conventional chemotherapy or otherwise, you're always going to get tumor resistance emerging because some of those clones will have found a way to escape. So people have found that they're able to link maybe 2 binding domains together, sometimes 3, although really that's getting kind of sketchy. So you're very much limited in having to recreate a new synthetic antigen receptor for every single target you have. And we already know there's many, many, many potential targets out there, as we've seen from all the antibody drug conjugates and other antibody drugs. There are many targets we can go after. And so that would mean that if we're going to have CAR T cells or SAR T cells against these targets, we're going to need many, 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 many of these receptors. And they're not inexpensive to test and evaluate and manufacture. So enter the idea of a programmable synthetic antigen receptor. And that's, this is not an idea that I myself have come up with. It's been out there for a while. The traction's been modest, but it's got a very interesting idea. And so here, you would only create one synthetic antigen receptor, one and one only. And you would allow small molecules to direct that antigen receptor against tumor targets by having an acceptor domain on your synthetic antigen receptor that can bind to a donor domain. And very often people are using things like FITC or PNE or other kinds of well-known molecules. And then you'll have molecular adapters, which could be any number of ligands for tumor targets, small molecules, proteins, you name it. And what's attractive about this is you manufacture one large prep of T cells, you can infuse the patient with those T cells, and then you can, in dynamic fashion, modulate what these T cells can see by infusing the patient with different molecular adapters. And so this is an interesting idea and one that I've been exploring in my lab with a colleague who is a chemical immunologist. So at this point, I'll be sharing with you some recent data from our group just to tantalize you a little bit. And I want to acknowledge the folks who've done the work. And it's really been Nick Cerniak, who is a chemical immunology student in my lab, Ali Moore, who works with him, Bonnie Boyevich, who provides him support, and thank the Samuel Family Foundation for having given us the cash to take some chances. So my colleague Anthony Rouleau has developed this technology wherein he can covalently bind whatever he wants to antibodies. And he does so by targeting antibodies with antigens that they recognize, and then using covalent chemistry to trigger a reaction that will ligate this ligand bind domain shown here in red to the antibody. And his interest was really redirecting antibodies against tumors, and recently he's redirected them against SARS-CoV-2. But it's a pretty attractive way to modulate the antigen specificity of an immune receptor. And so that got me inspired to ask the question of, could this technology benefit the universal SAR strategy? And so we asked a very simple question. If we generate a ligand binding molecular adapter that contains a covalent group, would it be any better than one which doesn't contain a covalent group? So we created a synthetic antigen receptor based on the TAC platform in my lab, where we introduced an acceptor domain, and we asked the question of whether we can label T cells engineered with this synthetic receptor with a model small molecule adapter where biotin was in place of the tumor ligand bind domain. And then we could detect biotin on the surface of the cells. And to summarize these data very quickly, by flow cytometry, the only molecular adapter that could bind effectively to the synthetic antigen receptor was the one that bound covalently. So here we have an example of what you see when there's no adapter at all, a non-covalent adapter. Here we've got the covalent adapter in the presence of excess of this donor molecule. Here's the adapter alone, and then of course other controls in the presence of the adapter. So the adapter will only covalently bind to the T cells in the presence of the synthetic antigen receptor. So what does that mean in terms of functionality? So here's an experiment where we put everything together in a dish. We've got tumor cells, we've got T cells that are engineered with this, what we call the acceptor TAC or the ACTAC, and then we add in a very limiting amount of the covalent immune receptor or non-covalent, and you find that tumor cells growth is completely suppressed in the presence of the covalent immune recruiter, but the non-covalent one is unable to mediate much in the way of suppression compared to the T cells alone. So we're seeing a real benefit to covalency in terms of triggering attack against the tumor. And in this case, we've used a small molecule ligand called glutamate urea that binds to PSMA. Thinking about delivering these things in the long run and the high expense of autologous therapies, we were inspired to try and create an off-the-shelf solution based on this technology. And as I mentioned at the outset, conventional T cells that recognize MHC peptide require autologous manufacturing because they come from every individual separately, and you can't take T cells from one individual and put them into another because you can get graft versus host disease. And so it's difficult to use conventional T cells as a source of off-the-shelf products. But there's a subset of T cells known as gamma 9 delta 2 T cells or gamma delta T cells that don't actually recognize MHC peptide. They recognize conserved molecules. And it's this alter recognition that allows these cells to be transferred from individual to individual. So we're currently exploring the use of these gamma 9 delta 2s as tools for an off-the-shelf product. So we rationalize that if you engineer gamma 9 delta 2s with one of these universal synthetic antigen receptors, we could have potentially a universal off-the-shelf product that could be tailored to the needs of the patient and then reprogrammed in a dynamic fashion, which is exactly what I said over here. And so does it work? The answer is yes, it does. So here we didn't want to prejudice the field by only studying T-cell antigen couplers. We also studied chimeric antigen receptors. And in both cases, the results are the same, that if you engineer gamma-9 delta T cells with universal synthetic antigen receptors, regardless of their platform, be it TAC or CAR, they both show the benefit of covalency, shown here by the open circles in the case of the TAC or the CAR. And here we can see with the non-covalence, there are some anti-tumor effects, but not nearly as robust as covalency. So clearly the covalent chemistry is playing a really critical role in allowing us to effectively modulate the functionality of these cells by redirecting them at tumor targets of our choice. Another interesting point is that we can get down to concentrations in the low nanomolar range, which means that this is a pretty potent tool that allows us potentially to use concentrations that we could achieve in humans. And I'll just note that although we've been focused on small molecule drugs to this point because of their flexibility and because of their bioavailability, we're also exploring the use of other ligand bind domains because we believe that all ligand bind domains should be amenable to this strategy. So, and of course, with any synthetic antigen receptor. So pretty exciting opportunities, we think. Just to summarize what I told you, the programmable synthetic antigen receptors can overcome this rigidity of conventional receptors that are limited to single or maybe two targets. Covalency really improves the performance at limiting concentrations. And the other interesting thing is this technology opens up the opportunity to use ligands that cannot be genetically encoded. Say, for example, aptamers, which are very interesting ligand bind domains, but you can't possibly introduce that into a cell through engineering. So I'll end it there and be happy to chat about this or anything else in a moment.